Thank you for coming to this session. My name is Alpa Patel. I work at Nelson Labs. And for the next 40 minutes, I'm going to talk about reusable medical devices and how we perform validations on that. And hopefully you guys can learn something about the materials that are used here. I've been walking through this platform, looking at all the different materials that are used, and a lot of them are constructed for single-use devices, but many are also used for reusable. And when I talk through the validation strategies, it will give you an understanding of how these raw materials or even little parts that are used to make a reusable product, what do you have to consider? Now, when I talk about reusable, I'm talking about a lot of people assume these are devices that are used on patient. Yes, these are devices that are used on patient, and they are used repeatedly. So if it's used on one person, it gets cleaned, disinfected, and sterilized and used on a second patient. And this happens until the device breaks down. Now, you might say, when does this device break down? Is there something that we know or we have to test for? Recently, there hasn't been anything, but I will go on and you will see in my slides when we're starting to look into stress testing and repeated use of these devices, and when is the break point of the materials that will cause a safety problem when being used. But I also wanted to bring an emphasis is when we're talking reusable, we're also talking about non-critical devices. These are devices that that are not patient contacting or they are patient contacting, but it's only with intact skin. So during the presentation, you will see that I do cross over from critical to semi-critical to non-critical, and these are different categories of devices that are utilized for validation plans. And why am I focused on cleaning is because these devices here that you'll see the yellow spot on, they have hospital practices, right? There are organizations in healthcare facilities that do write procedures to clean these type of devices. However, regulatory has now enforced the manufacturers that make these devices or any devices that are assisting critical procedures. Now, if you think about a robotic device, I'm sure you've seen some here that are doing the surgery. The equipment that is helping these robotic devices in the procedure are the non-critical devices. So they need validations as well. They have to be cleaned and disinfected. Yeah, they can be sterilized, but there has to be some sort of a decontamination process established to ensure that there is some process that you have validated that the healthcare facility may or may not use. We all know that healthcare facilities have their own clinical practices and organization committees that do write decontamination procedures for how to process these. But you also have to have a validation. And to give you perspective, you will see a surgical light here. And when we got it in our lab for testing, the first thing in my mind was like, why are we testing this? Do we really need to do a validation on it? And how are we going to do? The light was literally half the stage, right? So I had one light in every lab. And trying to do a cleaning validation, so you can assume that I'm going to contaminate these lights with artificial blood, right? I'm going to spray on it. And now I have to clean these things, and then I have to disinfect them and put bacteria on these and try to get 1 million bacteria dead in 1 minute. That is what we have to do, right? Is that possible? No, not really. So we had to come up with some strategy of how we're going to make it work. But the manufacturer of that light had to prove to the FDA that it can be reprocessed. They needed to have a procedure, and it had to be validated. And it is non-critical, non-patient contacting, but it is a cross-contamination zone where you can infect a patient, and that's why it's considered for validation purposes. And stools footstools, we're starting to see those come in as well, wheelchairs, anything that can be used for a patient that is non-critical. We're starting to see a ramp up. When I've been in the industry doing these validations for 20 years now, and just the last 5 years is when we have started to see a ramp up for non-critical devices, and we're seeing more and more these things that are in the patient's room for moving the beds up and down, right? We're seeing validations for those. We're seeing validations for TV monitors, remote control that's in the patient room. So we're IV poles, like I said, bed poles, mattress covers. All of that is something that we're starting to see come in is validations. And the reason I'm bringing into this platform is because a lot of these devices are not meant to be reused with chemicals that are harsh. And cleaning is fine because I can wipe and get it off and show that, you know what, there was blood on here, I got it off, we're good enough. That's the acceptance criteria. Visually clean, good. Disinfection is where we're having issues with, because the wipes that are sold in the market, Cava wipes, IPA, bleach, you've seen those wipes that you use at home, same type of wipes are being used in the hospital as well. Now, those wipes on materials can degrade so quickly, and that's why we're seeing a lot of repeated testing on these products as well. So just not critical, but these as well, to see how long does it take before we start to see a stress crack, or we start to see the polishing and labeling all gone. And with the MDRs coming up, right, where we have to have a UDI number on medical devices, these are some of the things that you will have to do testing on. Are my labeling still there? Because they will have to scan these devices in. Now, I'm going to go over the roadmap of how these cleaning validations, and I'm focusing on cleaning validations because other validations like disinfection and steam sterilization are very easy. There are criteria out there. You know exactly what to do. You know what is needed and how to perform it. It's the cleaning side of the world that people they don't really understand how those validations are performed, and that's why I'm here to talk about just these validations. But I'm happy to talk to anybody after this talk about disinfection or steam sterilization as well. Most of reusable medical devices, 80% is usually steam sterilized. Some are low temperature sterilized, EO, VHP. There are other modalities out there that can be used, but this is where the bread and butter is when you are working with reusable medical devices. If you can't make this work, you cannot make disinfection or sterilization work either. Okay, this is the first step in any reprocessing, is you have to have this validation. And just recently, in August, AAMI published AAMI ST98, which is a cleaning validation document, the only document in the world that actually now has instructions on how to do a cleaning validation and what you need for that. Now, the first thing you want to do when you are thinking about these validations is product categorization. Where does your product belong? Is it critical? Is it semi-critical? Critical are devices that go into your bloodstream, right? So it is getting in contact with blood. Is semi-critical devices, these are devices that are going into mucous membranes, right? ENT devices, ear, eyes, nose, throat. Those are the devices, bronchoscopes, that's what we're talking about. Non-critical devices, I just showed you the whole slide on that. So those are the validations that, depending on what your category is how we would draft a protocol, right? We never write a protocol before we know what your device is and how it's being used. So that's the first thing we need to know, is what is the category and how are you submitting your 510K with a category. And then we start writing a protocol. And the protocol alone is a long transition. A lot of people think that protocols are, you just tell the lab this is it and you move on. Our longest timeline is on the protocol development. The testing is fast. It's usually a month. We've had protocols for 6 months still figuring out how we're going to write the validation plan, because it depends on how the product is being used in the healthcare facility is how we are writing this protocol. So we're asking engineers or people that are calling us for validations, how are these devices being used? How are they being contaminated? How are they being manipulated? Have you seen the central processing department that does all the cleaning? How is that being performed? Is your device capable to withstand ultrasonic cleaning? Can they withstand high pressures, high temperatures in automated washers? Can they deal with alkaline detergents, pH 13, or is it going to break down in one-time use? Or is it, those are the things that help us put a protocol together, right? And so that does take the longest time. And then we do the execution of the protocol, and then we do the cleaning validation, where we are doing the test soils and figuring out the method of cleaning to make your device work better for the category that you've chosen. Then we're testing for analytes. So we're not testing bacterial for cleaning validation, okay? Back when I started 20 years ago, we did only bacterial. And then we had analytical markers where we decided to do protein and hemoglobin and TOC. And you may ask why? Why not bacteria? Because that is usually the source of cross-contamination or infection rates in healthcare-acquired infections that happen. The reason for that is because cleaning is the first step in reprocessing, and then it's followed by disinfection or sterilization. So disinfection and sterilization are taking care of the microbial load already. That's why we don't do microbial for cleaning. For cleaning, we're looking at analytical tests where we're figuring out protein, hemoglobin levels that are left after. Why? Because that is a food source for microbial growth. If I have anything left over, that even if it's water, bacteria loves water, right? So even if I have a little bit of water, that is where I'm getting my bacteria growth if I don't dry it appropriately, if I don't challenge it appropriately and remove the proteins and hemo off of it. So that's why we do analytical tests for cleaning, and then steam sterilization and disinfection after they will take care of the microbial load at the after effect. Now, this is the industry market for reusable medical devices. A lot of people get surprised when they look at this chart because it is something that a lot of people don't realize, that all of these instruments are reusable, and we actually we validate all of them. And we have surgical instruments, orthopedics, huge, those big, huge surgical trays that you built. Those are the trays that we're validating that can be used, right? Do we validate every single one of them? No, we usually pick out the worst case or challenging devices out of the tray and validate it that way. So if you can clean that, you can clean the rest. ENT devices, bronchoscopes, endoscopes, all of the flexible endoscope, dental. We're starting to see dental regulations push the dental industry for reusable devices. They never did before. Dental was something that was slid and nobody knew that they existed for reusable. We're starting to see that those are coming up now, and they are being challenged by regulatory for validations. You're using this ultrasonic unit to clean. You've gone to the dentist. You know how they just throw it all in that ultrasonic bath and they put it in there and they start it on. We all know that when you layer instruments up, you're not cleaning anything. So those are the things that they are now challenging and asking questions about. Robotic devices, the next generation for innovation for devices, right? We have so many robotic devices right now in innovation, in stages where they're trying to bring it out in the market. And validating those, I'll say, is a beast, right? Trying to validate those devices where they have those surgical forceps or monopolar bipolar forceps that are so small and you can get soil stuck right in, but I don't have a flushing point to flush them out. Those are the challenging features that we work with for these devices as well. But that is a market that we're starting to see creep up as well. Home care products, all of the baby monitoring, baby devices, that baby bottles, baby sanitizers, all of that, we validate all of those things as well, right? Diabetes monitors, they all have to be validated and should have a validation instruction for them. Cardiovascular devices, see them all the time. And then we also do dialysis machines. This is the last 4 years we've seen an uptake on dialysis machine validations. So everything is single use. You pull the tubing out, you throw out. But what about the internal tubing, right? That still is intact. And there's water in those pipelines. So now we have to figure out how we're going to kill. If there's water, there's Pseudomonas, there's bacteria growth. That's a fact. So we don't want to cross-contaminate the chemical that's going in there. So that's why we're starting to see an uptake in that as well. So dental and dialysis machines are two things that we're seeing aggressively by regulatory, pushing for reprocessing validations. This is just an example slide that I put together. So it gives you an idea that when I talk about different levels of critical, semi-critical, or non-critical, what type of devices I'm looking at, and what type of method is used. And usually you would see for non-critical devices, a wipe is done and it's fine. That's fine. For semi-critical, there are ENT devices, you need a soak. And for critical devices, it's sterilization. Okay, usually steam sterilization, or it's what was I going to say? Low temperature sterilization. But you can also use high level disinfection. But if you ever do that, you have to show regulatory that you did try to use your devices in other modalities and it just wouldn't work, and that's why we're doing high level disinfection. They will require some sort of justification on why your critical device cannot be sterilized if you do choose to go that route. Now, protocol development, I think we talked about that earlier, but it is a collaboration between a manufacturer and a testing lab. For FDA submission, as a testing lab, we understand what is required for those validations. As a manufacturer, we expect that you understand your devices that are coming to us. We don't understand the devices. A lot of times, our study directors don't even know what the devices are used for, right? We just go on YouTube and try to search for the name and see if there's any videos that we can see so we can establish a protocol out of that. And depending on the criticality of your product and how it's used is how the acceptance criteria will be set up for the validation as well. Now I'm going to go into cleaning validations a little bit more in detail, but these are basically the 5 steps that are involved, right? We have simulated use cycles. We soil the samples with artificial test soil. We clean them per your IFU, your instructions for use. If you don't have an IFU, we can always help you make one or develop one that you need for. We extract those samples and then we test for analyze. Those 5 steps, right? That's all you need. Now, simulated use cycles, the reason why we do simulated use cycles is because of this. When I first started, we never did that. We would get brand new devices from manufacturers and we would do validations on them. And yeah, they'd pass with flying colors. They looked great. We didn't have to worry about it. Over time, in 2015, when the FDA guidance came out, they started the simulated use testing requirement, right? It was a recommendation, but if an FDA puts out a standard, we all know it's a requirement. So that's when they started mandating simulated use, and it was for good reason. A lot of recalls happen, and when the recalls happen, that is what we see, right? Retention of soil, retention of debris in the devices that have been left over that we couldn't clean, but they're sterilized. And once you sterilize tissue, you're not getting that off no matter what you do, right? So it's going to stay there in forever, and it's just going to accumulate as it goes. And that's how healthcare-acquired infections rose up, and they started looking into this. Over here, you can see that's a surgical suction cup that University of Michigan did a study on. They opened up all of them, and that's what they found in there. So that was an awakening for all of us to figure out. Now, why I have this picture in there is because back when we did the validations, I did that validation. I validated the suction catheter, right? Now, with the validation, I did not challenge it with the tissue. Why? I didn't know that it suctioned all the tissue in there as well. So working on the protocol with the manufacturer, we missed that critical part. Does it come in contact with blood? Yes. Checkbox, let's do that. So we did that. We put blood, but we didn't put bone, we didn't put tissue, we didn't put bone cement that orthopedic devices often see. Now, it is a requirement that when we are doing these validations, we have to actually see the procedure to figure out what we need to put in. And it is heavily challenged. Anything that uses electrical surgical instruments, we have to have the machine there. And we have to test them with the electrical surgical to char the blood and tissue onto the product, and then do the reprocessing. Now, over here, you'll see that this is a study that we did with rusting inside the instrument, okay? It was a suction catheter as well, but it rusted. It passed all the cleaning testing, all the markers, all the protein, hemoglobin, everything passed, but cytotoxicity failed. And when the cytotoxicity failed, they called me down to the lab and they said, there's something wrong. We can't make this work. Everything is fine. The customer is upset. Can you figure this thing out for us? So I pulled out a boroscope, looked right in there, and this is what I found. And as most of you guys are aware, if you have rust, you're failing cytotoxicity. And it ate up the insides, and that was only 6-time use. This device would be in the market infinity. Outside it looked great, but if you're not looking inside, which is not a requirement for reusable devices, we're trying to get it, but it's probably not going to happen because healthcare doesn't have the time to look at every instrument that they process, right? They just pick one out of the 50, look in, looks great, move on. So we do these simulations in-house now to make sure that at least we've done the 6. Now, the AAMI ST98, this is a new standard that just came out in August. I will be talking about the standard specifically tomorrow during my talk. And there's a lot of changes for cleaning validations that have come up that if you are in the reusable end of the world, you probably want to know these changes right now because regulatory is enforcing them. Usually there is a grace period, but it depends on the reviewer if they give you the grace period or if they don't. We're seeing that they're now implementing already. Now, the 6 simulated use cycles, which is the first part of the validation, that is taking the device into the entire processing that happens in the hospital. You use it on the patient, you clean it, you sterilize it, you shelf it, you do it again. So that's what we're doing 6 times. Now, you might be like, why 6 times? It's a number that came out from the FDA. It was 10, it moved to 3, and now it's 6. And they're comfortable with 6. We have seen that Europe has actually asked for these testing after the life cycle has been performed. So if you say a device is good for 200, they want to see 200, and then you do these analytical tests to make sure that they're still safe. Not everybody, but we've seen notified bodies now asking for that. In Europe, not here, but there. These are sample size controls, way more than any other tests that we do. We have more sample controls than we have for any other test because we're working with analytical tests for determining protein and hemoglobin and TOC and all of that. But you need to have a certain amount of devices for validation. They don't write how many in the standard. They are asking that the manufacturer tell us what is the right sample count for the validation. The standard that I'm going to be talking about tomorrow does allude to 6 is appropriate, but depending on the criticality of your device, that number will fluctuate and change. As for positive controls, the big impact that happened in August was we only did 1 positive control for these validations. Now it's required that you have 3 positive controls to show reproducibility of your extraction that you're performing. The industry got away with 1 for a very long time, but I think FDA has caught up that to show reproducibility of the validation and assay, you need at least 3 samples, even if it's a positive control. Now, selecting test soils. First thing we're going to ask the manufacturers, how does your product, what does it come in contact with? Is it blood? Is it mucus? Is it tissue? Is it bone? What is it? And from there, we go on and validate what is needed. The ISO documents and ASTM has categories of where your device fits in and helps you select what test soil would be appropriate for your product. Coagulated test soil is mostly used in Europe. FDA likes to see coagulated or defibrinated. That's what we use. And mucus test soil, the British test soil, that's what we use for mucous membranes. This has been standard for the last 20 years, right? What we do is we just add things to it. Let me give you an example. When we had what am I going to say? Liposuction canisters. Those were thrown away, single use, right? And the manufacturer said, hey, we want to make this reusable. Can you clean this device and disinfect them? And I said, I don't know about cleaning. It's got a lot of fat in it. And they're like, well, can we try it? I said, okay, can you send me what does this canister contain so I can make a test soil that looks exactly like what you're collecting from the body? And he sent me pictures of it, and I looked at it, and I was like, yeah, I think it's doable. So we got sheep blood. I put Crisco. I blended it together, and there's my test soil for the liposuction canister, right? I used that to soil the inside and some outside, cleaned it, and it was fine. It's reusable now. So we do work with every device is like a custom protocol. And so we don't have a standard test soil that we will use. We will look at your product and be like, okay, it comes in contact with this. We may have to mix this soil and this soil to simulate that. If it's going colonoscopes, we may want to use a fecal test soil. We've used that before, fecal test soil with some blood in it, and use that for the colonoscopes. So things like that, we always are trying to figure out what is the best simulated test soil to use for these validations. This is a chart that regulatory likes to see all the time, and it's clinical relevancy. So if you use any test soil, they're not going to accept that. That's an automatically red flag, right? You want to make sure that the test soils that you're using are clinically relevant, just like I talked about. How does it represent your device? And how are they coming in contact with them? So our protocols will always have this chart in there. So when you submit our protocols to the FDA, they have this already embedded in there, why we chose the soil and how they're clinically relevant. Now, soiling methods. When we do soiling methods, that is one thing that we also ask manufacturers to work with us is how are these devices soiled? And we look at the process and then we soil it to the worst case. FDA and all regulatory agencies require us to test at worst case, just in case something does happen wrong, then we have incorporated that into our validation plan. So we will test it worst case. Scissors will never look like that, but we would still test it at that category. So that is how we would do. Now, there are soiling methods that we do. There's flushing, handling, like you just saw. There's immersion, that's down there, and spraying. And spraying, like I give you an example with the surgical lights that we have. When you cut if you cut a vein or artery, it's going to spray up. That is what we're looking at. For non-critical devices, it's usually a spray or handling, basically. Time for drying. So once we soil the devices, they're going to sit there until they're dry. Where all the IFUs say clean immediately, that's a dream, right? That's not going to happen in a healthcare facility. It goes down to the central processing departments, and they will clean at their time and when they have the ability to get it done. And the picture I have up here is a real picture from a hospital that we visited. And these are trays that are sitting to be reprocessed. So if you think about it, they've been sitting there for 8 hours. They have to clean all this in 8 hours. So you can see how fast they're going. If you don't clean it in 8 hours, you're gonna be in trouble, right? So they're just going as fast as they can, flushing, brushing, whatever they need. But every device in a surgical tray has to be clean. If you think about a tray, there's probably 30 devices in each tray. And then you have soiled product in here that's going down underneath your tray, or it's trickling down. And these have to be all washed within that 8-hour period. And then the next shift takes on. And so that's why we challenge it until it's dry to the touch, to challenge the cleaning processing. So for the cleaning, we have manual, automated, and mechanical cleaning. So a lot of manufacturers will say, my device cannot withstand ultrasonic cleaning, so we're not going to validate that. The reality here is healthcare facilities will put it through an ultrasonic cleaning, regardless you write it in IFU or not, because that is their common process. Right? So it's very important as engineers to understand what is happening in the field that you are selling this product to. Because when I went to audit hospitals, that's what I saw. Rinse, bath, ultrasonic cleaning, straight into the automated washer. Right? And that is how it's being processed. And once it's being processed that way, regardless what you write in IFU, they're not following that. They're following their internal process for it. So just to bring that picture in your head that if you are making a reusable devices, you probably want to make sure what is the common process that's going to be happening in the healthcare facility as well. Validation parameters, we're not going to follow exactly what your IFU specifies. It is required that we do take steps, we cut steps out, or if a detergent says soak 1 to 4 minutes, we're doing 1 minute, right? What is healthcare doing? They're saving cost in detergents. They're not going to do five ounces per gallon. They're going to do 1 ounce per gallon. And so we're taking it to the lowest range. The lowest range. The time is essence. We have to make sure that when we're doing these validations, that we're not taking 10 minutes. We're taking literally 5 minutes, because that's all they're spending their time on, is 5 minutes, and then they're moving on. So if you write 10 minutes, it's not happening. So we will take it down to 5 minutes, or we are always doing default settings for washers as well, just to make sure to encompass that if they do worst case, we have validated that process and we're okay to go. They can go above if they want, but as long as you've done the lowest dose, temperature, and range, we're fine for the validation. This is an example of what an automated washer looks like. I've got thermal grade out here because we do not use that for validations. And you have to validate that separate as a disinfection step. Although it's processed in the healthcare facility as the same step, it's not for validations. We have to take certain steps out. That is one step that we have to take out. Detergents, this is a big one for materials. We often see that when we do alkaline detergents on these products and we're washing in ultrasonic cleanings, they will break down way faster than the enzymatic neutral pH detergents, right? The neutral pH will make it work maybe 500 times, the alkaline 200. So there is a huge significant difference. The 10, 15, 20, you don't see anything in that range. You see it in the higher ranges where you start to see material break down very fast. You will also see hard water buildup on the alkaline detergents, and you will see the white film on your product. And that is, oh no, you can't use that anymore. They'll trash that out really fast. So it's quite important to know that you're doing that. Now I have here, do healthcare facilities use detergents you validated? No, they don't. They do not. They use what they can find and what they are qualified to buy. Usually they're buying huge drums of what is the cheapest detergent they can find because they're going through drums and drums of it. We have our detergents that we have probably 15 of them that we talk to the manufacturers about. These are the bank of detergents we have that we can validate and use them on, but that doesn't mean that the healthcare is going to use what you validated. The last step after cleaning is visual inspection, right? Visual inspection is very important. Believe it or not, for an instrument that goes from one body to another body to another body to another body, visual inspection is only required. Nothing else is required. So you visually look at it and move it along. There's boroscopes, but it is a recommendation for lumens that you use a boroscope. But other than that, it's just a recommendation. And magnification with 10x, only flexible endoscopes, duodenoscopes, and high-risk endoscopes have to be inspected with 10x. Anything else, no magnification is required for that as well. So it depends on your device what kind of inspection we'll do, but majority is visual inspection is a requirement. Once it's that, we are extracting your product, and usually it's sonication or manual shaking, or flushing. So these are some of the extraction methods that we would use for taking if there's any soil that's hiding in the devices, we can pull it out and test for them. The acceptance criteria I have listed here. You only need 2 analytical markers. The protein is a must. Why? Because protein is in every building blocks of every cell. So you have to test for protein. Hemoglobin and TOC are the next 2. Depending on what your product is what we would advise. If it's blood contacting, automatically I would do hemoglobin. If it's ENT devices where it's going your nose, your ear, your eye, I would do TOC. So that's how we break it up. But you only have to do 2, but they do write a whole list that you can choose from. But protein is one that's required. And we don't do any bacteria like I talked about before, because the next steps will take care of the bacteria. These are the acceptance criteria outlined in AAMI SD98 that was published in August of this year, and ISO 1583-5 that was published in 2021 in February. So you can see that Europe and US don't meet. They don't see eye to eye. And so FDA does not recognize ISO 15883 series, and Europe does not recognize AAMI SD98. So as a manufacturer, now you're in a pickle over here, trying to figure out what am I going to validate. But if you see that the action level and the recommended level match, right? But we're seeing that notified bodies in Germany and France are actually looking for 3 micrograms per centimeter squared for protein. So just to be aware that they are using the ISO 15883-5 acceptance criteria for their devices. If you haven't looked at this standard, it is one that you probably want to be familiar with. This is the FDA guidance standard. It talks about everything I talked about today, including cleaning validations, disinfection, steam, repeated use, and packaging. Everything is in here, what you need to do for a reusable medical device. And they use this as a backbone for when you submit your 510K submissions to look at if you are following what they have recommended for testing. And so we use this as our reference to write validation as well. This is a new standard that got published that I just talked about, and I will be talking about this tomorrow in detail on what the new standards are and how are they going to affect cleaning validations, because it is a big change, and it's happened after 10 years, and I think it will be something that if you're working in the reusable, you want to know what these changes are. That takes me to my last slide. If you guys have any questions, I will be happy to take them anytime right now.
I was wondering if you could clarify which standards the FDA recognizes and the EU. You mentioned the AAMI and the other.
The ISO?
The ISO, yep.
Yeah. So FDA recognizes a lot of standards with EU. However, the ISO 15883 series, they don't recognize that one. But they do. And a lot of standards, if you look at the first 2 pages, they go through what documents their consensus with. So all the documents will have that. But they do recognize the ISO 17664, 17665, EN 285. So they do have those, but the ISO 15883, that is a washer disinfector document. That is one that they don't recognize. Any other questions?
Hi. Do you foresee the FDA cracking down on cleaning validations for implants as a source of contamination?
For implants, we don't see that, but because it's one-time use, and that's it, because it is implantable. However, there are some implants in surgical trays that you have that they will pick one out and the rest are there. A lot of manufacturers say throw it out and repopulate. But if you do not say that, they're going to reuse those. And when they reuse those, you don't have validations on those. And we see those come in for validations. They haven't hunked down on that yet. But they know that this is happening. So I've seen a handful of those validations in my 20 years, but like maybe 10, but nothing more than that.
I have a question on the drying. So when you were talking about like the clinic validation, you mentioned that it is important to assess it when it's dry to the touch. Do you do a lot of validations with like accelerated drying? Because I'm just thinking about the time that it would take to get to that point. What are your kind of like comments or challenges with that?
That's a very good question. So with accelerated drying, it will challenge it more. It is not realistic. But do you mean accelerated meaning if it was 24, I do it to 72 hours or both? Okay. So we have done 72 hours. This is when when the hospital is sending your device out to a third-party processor to clean. So it's going in a car somewhere else to be cleaned and come back. That is when we do 72 hours, right? We're doing studies right now, and Amy's writing a document TIR 109 just on that after 19 hours of test soil, the polarity of protein changes, and it becomes hydrophobic. So it's hard to take it off. So these are the studies that are being processed right now for the Amy documents that we're writing. And it may be that FDA might say it's not to touch anymore. You need to do it to at least 19 hours or 20 hours. But 72 hours is the cutoff for dry time usually. Well, I hope you guys enjoyed this session. If you have any other questions, I am in booth 3418 if you have any questions. Thank you.